Hello, everyone, and welcome to this session of ACCB 2020. This is a joint session of papers in the genes and proteins track. My name is Ana Conesa. I'm a professor at the University of Florida, and I will be chairing this session. So please remember that if you have questions, you can use the Q&A chat. You will not be able to speak, but I will read these questions. And if you can also vote on the questions of somebody else, so they will get prioritized. So every speaker will have 12 minutes and then 2 minutes for questions. So without any further ado, I am very happy to introduce you to Ayse Dincer. She's going to talk about adversarial deconfounding autoencoders for learning robust gene expression embeddings.
Thank you very much, Anna, for the introduction. Hello, everyone. I'm Ayse. I'm doing my PhD in computer science at University of Washington. And today I'm going to talk about my project, Adversarial Deconfounding Autoencoder for Learning Robust Gene Expression Embeddings. Okay, I think it's a little slow, but it will come around. Okay, awesome, perfect. So as you would all know, gene expression profiles provide a snapshot of cellular activity, which might enable us to understand various diseases better. And as the amount of expression profiles that are available is increasing, we are now able to use complex nonlinear models on these expression data for various biological problems. And I believe that among all these models, unsupervised deep learning has enormous potential to extract important signals from vast amount of expression profiles. And as a super quick reminder, an unsupervised neural network, or autoencoder specifically, consists of an encoder and decoder network with a bottleneck layer in the middle. So the goal of this network is to learn an embedding such that it can reconstruct the original input features as successfully as possible. And I believe that these models are very promising, first of all, because they give us the ability to train informative models without supervision, which is great because you would all know how difficult it is to come up with a large number of samples with coherent labels. And the second reason is because we can extract patterns from the data without imposed directions or restrictions, which can be the key to actually learning something new about the biology. So it sounds very promising, but unfortunately, it's not that straightforward to employ these unsupervised models on gene expression, because expression measurements often contain other sources of variation in addition to the true signals we seek. For instance, we can see some technical artifacts not relevant to biology, such as batch effects, or we can see some out of interest biological variables, such as age or medications used. And we call these biological or non-biological artifacts that are systematically affecting expression values confounders. And the problem with confounders is that for many existing expression data sets, these confounder-based variations can mask the true signals and prevent us from learning meaningful representations. So just to give you an example of that, here we have some expression measurements from a breast cancer, and we're combining two different studies. And we simply plotted the principal components of this expression matrix. And you can see that the two data sets are clearly separated, showing us an example of dominance of batch effects. And then we simply took this expression matrix and passed it to an autoencoder and generated the exact same plot, this time for the autoencoder embedding. And you can see that this dataset variation present in the original space directly transfers to the latent space as well, which is quite problematic. And moreover, another problem is that confounders can also prevent us from learning a robust transferable model. So we want to generate embeddings that can capture biological signals that are valid across reasonably similar domains. And to just show you an example, here we trained our autoencoder from the previous two data sets and encoded samples from an external test set. And again, when you look at the principal component plot, you see that the external samples are placed quite separately from the training samples, just giving us an example how these confounders create problems in terms of transferability. So in our project, we address this problem of entanglement of confounders and true biological signals. And our goal is to basically show the power of unsupervised deep models by generating biologically informative expression embeddings that are robust to confounders and also are generalizable across different domains. And to achieve this, we propose a deep learning approach, which we call adversarial deconfounding autoencoder, or ADAE in short. So now let's delve into what ADAE actually is. Our model actually consists of two subnetworks. The first one is an autoencoder that learns an embedding such that it can reconstruct the original input features successfully. And the second model is an adversarial predictor that takes the embedding generated by the autoencoder as input and predicts the confounder variable. So the goal of this entire model is to learn an embedding that encodes as much information as possible while not encoding the confounding signals. And the way we achieve this is we apply a joint training. So we first pre-train the autoencoder to minimize the reconstruction loss, and then we separately pre-train the adversary to minimize the adversarial loss. So here, by the way, the adversarial loss is defined based on what type of confounder you have. And the most important part is we move into joint training where we freeze the adversary for one epoch and then train our autoencoder to minimize the joint loss, which means minimize the autoencoder loss while maximizing the adversarial loss. And then we freeze the autoencoder and train the adversary for one epoch to minimize the adversarial loss. So basically this competing alternating training process is for autoencoder to learn an embedding that is as successful as possible while preventing the adversary from doing a good job, but the adversary is still trying to improve its performance. So when we train these models together, they will get optimized to learn an embedding that is informative but free of confounders. And the last point is, as you can guess, this lambda variable here determines the ratio between the deconfounding and the reconstruction. So you can increase this lambda to get a more deconfounded embedding. So in our project, we just wanted to show the broad applicability of our model by applying it on different expression data sets and by different cases of confounders. And the way we evaluate our embeddings is, first of all, we check how well they can deconfound the latent space, how much they can preserve important biological signals, and how they can transfer across different domains. So just a small note, by the way, for this presentation, I'm only going to focus on our experiments on K-unplugged breast cancer data, which combines multiple studies, and we use the dataset label as the confounder. But we have many other experiments I would love to share later. So first of all, we want to show that ADAE can learn biologically meaningful deconfounded embeddings. And for this, we generated an embedding with the standard autoencoder as well as ADAE model. And we just created UMAP plots for this and colored them by the dataset labels. So here you can notice that for the standard autoencoder, the data sets are clearly separated, but ADAE can successfully fuse data points from different studies, basically showing us how we can deconfound with our approach. But more importantly, we colored the exact same plots, this time by different biological variables of interest, such as ER label or cancer grades. And here again, we showed that for the standard autoencoder, the samples are not that clearly separated by these interesting phenotypes. But for ADAE, we can clearly separate these different dataset samples by both phenotypes. So this basically shows us that once we remove the confounding effect, then it is more possible to learn important biological signals. Another property of ADAE is that it can learn embeddings that are generalizable to different domains. And to simulate that, we did a simple thing where we just trained our model on 4 data sets and just transferred to an external data set. So the embedding for the training is with the circle markers, and the external one is with the diamond markers. And here you can observe that for the standard autoencoder, the external samples are quite separated from the training samples, again indicating an unaligned manifold. But for the ADAE, the training and external samples are overlapping, showing us how we can match the manifolds. But the critical point here is that the direction of separation by cancer grade for the training samples is also valid for the external samples, which basically shows us that once you deconfound the space, you can learn more generalizable, more robust patterns, which is what one of our goals in this project. So I showed you a couple of qualitative analysis, but now I wanted to give you a couple of results from a quantitative analysis we did. We simply pitted predictor models on top of our embeddings and predicted various biological phenotypes. And we also compared in this way our approach to other deconfounding approaches. So here you see a small example where we predicted cancer grade on an external test set. Again, we train on 4 data sets and then transfer to an external one. And we compare our model to an uncorrected standard autoencoder as well as some other deconfounding approaches. And it was very nice to see that our model can significantly outperform the other approaches in terms of external test set performance. And I think a very interesting another result is that we also measured a generalization gap, which is the difference between an internal test set performance and an external test set performance. So this means that like if you have a low generalization gap, you can successfully transfer from one domain to another, which is, so we see that ADAE can have a minimal generalization gap, which is great because it basically shows us that you can transfer across different domains with minimal prediction loss, which is actually what we would want from a robot. So we have many other results on ADAE. For instance, we experimented on different data sets. We also experimented with different confounders such as sex and age. We carried very interesting transfer experiments. We carried very interesting transfer experiments like training on males, transferring to females, or transferring across different age distributions. And we even experimented with correcting for multiple confounders at the same time, like age and data set. So you can check out.
So you have 1 minute to go.
Okay, sure. Yeah. Okay, so I would love to talk about these results offline. So to wrap up, we introduced adversarial deconfounding autoencoder to generate robust expression embeddings, and we showed that we can learn deconfounded embeddings that can preserve biological signals and transfer across domains. For the future, we want to experiment with some regularized autoencoders, such as a VAE, for instance, and we also want to extend our experiments on correcting for multiple confounders at the same time. And most importantly, we just want to explore our model. We want to try different data sets, different cases of confounders, and most importantly, different diseases. So we're very excited about this project. I hope you're excited as well. And before concluding, I want to thank my co-author, Joe, my advisor, Xuan, and all the members of LeLab for their contributions. And thank you very much for listening.
Thank you very much, Ayse, for this clear presentation. Are there any questions on the chat? Okay, so while people are posting their questions, I have 2 questions actually. So one question is, it's not clear to me whether you provide information of the confounding factor somehow in the model, so that's some way this will be like a supervised problem. This is the first question. And the second question is whether you could use this approach also to integrate multiple types of omics data that are measured in the same set of features.
Yeah, thank you very much for the amazing questions. So for the first question, actually you're right. Yeah, the model is taking information about the confounder variables. So it is, if you have a confounder like dataset label, it is taking that. So in that sense, maybe we can consider it as supervised in the sense that it is taking some labels, but it is not taking information about like cancer grade or anything like that. But you're correct. For the other question, I think it's a great question. I really want to delve into that after this. So it can be possible by combining multiple models. You can also have model that jointly learns an embedding for everything. So I think that's something I definitely want to try. And for many different diseases, we have multi-omics data, and I think it can be a great setting.
Okay, thank you. So one question from the audience, from Claudio Diaz, who says it's a great talk. Is there any upper limit to the number of confounding variables? Thank you.
Actually, that's again a very good question. So I would say that it kind of depends on like the capacity of your model and the number of samples you have. So for instance, if you keep adding like different subnetworks for correcting for multiple confounders, then probably you would end up having too many parameters. And if you don't have enough like data power, then you won't be able to learn an incremental one. There are these kind of considerations, but I would say that it totally depends on your data and what kind of confounders you have, and maybe also even the correlations between different confounders that you have. So you might consider with one confounder network correcting for multiple confounders with just one network.
Okay. Okay. Thank you very much. Thank you so much. It has been a great talk. So we move to the next speaker, who is Hoang Pham, and he will be talking about his work on the driver group algorithm. So thank you. You can go ahead.
Hello, everybody. I will present a novel method for identifying cancer driver group named driver group. Oh, why is don't move? Yeah, a little bit slow. As you know, cancer driver genes are play critical roles in cancer initialization and progression. Therefore, there are many computational methods have been developed to identify cancer driver gene. Most of the current methods only identify single gene as cancer driver. However, there is evidence showing that in some biological processes, some genes work together to regulate the same targets, and in some cases, the regulation of one gene is not significant, but the regulation of group of genes is. Moreover, recently there are some wet lab experiments which have been conducted to investigate regulation of gene group in some biological processes. All of the highlight that there is a strong need to identify the cancer driver gene in group. And recently, there are some methods which are developed to study cancer driver in group. It's called driver gene modules. Suppose that we have a mutation matrix of the genes in a driver gene module of cancer patient at the figure. The idea of the driver gene module is that the mutation in only one gene in a module can initialize and progress cancer. For example, you can see in the figure, in the patient B2, only the mutation in gene 32 can initialize and progress the disease in this patient. Because of the nature of the driver gene module, the member of a driver gene module may not work jointly to drive a cancer. And one more thing, the current method for identifying the driver gene module only deal with the coding gene. But you know, non-coding gene also play an important role in the cancer development. So in this paper, we developed a novel method to identify the driver gene group. Yeah. A driver gene group is a set of genes which work jointly to impact on the target gene to drive a cancer. And as you know, proliferation gene related to the cancer development. So in our PRO method, we identify the driver gene group by detecting a set of gene group which work in concert to impact on the proliferation gene. And one more point is that besides the coding gene, we also deal with the non-coding gene. It means that this proposed method identify both the coding and non-coding cancer driver group. This is the overview of the proposed method driver group. I will discuss the detail of the method in the next slide, but in generally, there are three stages in the proposed method. The first one is constructing gene regulatory network based on the gene expression data of cancer patient. The second step is that discovering critical nodes in the gene regulatory network built from the first step. And when we have a critical node, we identify the cancer driver group based on the discover critical node in the second step. So in the first step, to build the gene regulatory network, we use the gene expression data of the cancer patient. Oh, it's a bit slow. Yeah, and each node in the network can be a microRNA transcription factor or a messenger RNA. The interaction between the two nodes is the Bayesian correlation of the gene expression of the two nodes, and the direction of the S follow the motif in the X figure. It means that a microRNA can regulate a transcription factor or a messenger RNA. A transcription factor can regulate a microRNA or a messenger RNA. A transcription factor or a messenger RNA can regulate all the transcription factor or messenger RNA correspondingly. And to just keep the real interaction between the two nodes, we refine the network with the existing database about the gene interaction, including the protein-protein interaction, the microRNA coding interaction, and the transcription factor microRNA interaction. In the second step, in the second stage, we discover the critical nodes in the regulatory network in the first stage. According to the sorry, can you move to the next slide? I cannot. It is really first one. Yeah, this one. According to the network control method, any network can control by a minimum set of nodes in the network, and the minimum set control the whole network is called the minimum driver node set. And we apply the network control method to identify the critical node in the gene regulatory network we built in the first stages. For example, we have a network like this, and we apply the network control method to identify the critical node for this network. And we find out the four nodes in the network with the red arrow is the minimum driver node set. So the size of the minimum driver node set for this network is 4. And after that, we remove node by node. For example, we remove the node with the dash border in the second one, and then we apply the network control method to detect the minimum driver node set of the new network. It means the network with one node removed. And we see that the minimum driver node set in the new network have a 5 node. It means the size is 5. And if the removal for a node increase the size of the minimum driver node set, we consider the removed node as a critical node. The reason is that when a node is removed, it increase the size of the minimum driver node set. It means that we need to interact more on the node to control the whole network. Say in another word, the removed node play a critical role in the network. And in the third stage, we identify the cancer gene group based on the critical node identified in the second stage. And to identify the cancer driver group, we create the combination of critical nodes, and evaluate the evaluation of the influence of the combination of critical nodes on the proliferation gene. And to evaluate the influence of nodes on the proliferation gene, we consider on the node in the combination of the we consider on the node in the combination of critical node, active node, and we use the diffusion model to activate all the node in the network. And the diffusion model have three rules. The first rule is that a node can be active or inactive, and during the diffusion process, inactive node can be activated by active node, but active node cannot inactivate it. And the process terminates if no more node can be activated. And the candidate cancer driver group are the combination of critical nodes which have influence on the proliferation gene. And before using the driver group to identify the cancer driver group, we evaluate the performance of the cancer driver group on detecting the regular effects of combination of critical nodes on all the nodes in the network. We use the data from the Dream4 Silicon Network Challenger. This data includes the 5 network, and each network have a 100 gene and 20 knockout gene pair. And we detect the target of the 20 knockout gene pair. We compare the performance of driver group with the performance of choice RDI and random method. As you can see the result in the figure, the performance of choice of driver group are performance choice RDI and random group in the first 4 network, and in the 5th network, the performance of driver group comparable to the performance of choice RDI and better than the random method. Next, we apply the proper method to identify the driver group for breast cancer patient, and we identify 28 protein driver group and 26 microRNA driver group. And the table shows the top predict driver group of the protein driver group, and you see there are many members of the predicted driver group are related to the breast cancer. And this is the top 10 of predicted microRNA predictor group. We also use the predicted driver group to stratify the breast cancer patient, and the results.
You have 1 more minute to go. Sorry.
Yes, thank you. And the results show that the survival of cancer patient are significantly different in cancer subtype, which are classified by using the predicted driver group. And the quality of the classification can be shown in the clustering display and the Siluter plot. And the last application for the proposed method is that we use the method to detect the driver group for the EMT, the epithelial mesenchymal transition. And we identify the 61 microRNA driver group which have an influence on EMT significance. And that's all about my presentation. Thank you.
Okay.
Yes.
Okay, thank you very much. I think we have time for a quick question from the audience. Anybody? Okay, I have one very quick. So how much is the computational cost of this? I mean, you are kind of comparing a lot of group of genes together. So is this computationally expensive, intensive?
Yeah, you are right. The computational cost may be a little high for the proposed method, but for my experiment with the breast cancer dataset, it's about 5,000. I just do some filter selection and I just select about 5,000 coding gene and 1,000 microRNA. And the time to run the experiment for this about 1 hour.
Mm-hmm.
Yeah, so it depends on the input data, but
yeah.
I think it's still the acceptable time for the method.
Okay, thank you very much. Thanks.
Yeah, thank you.
Thank you. Great, so let's move now on two talks from the proteins section. We have Wenjing Xuan online. Do we? Because he was not here at the beginning of the session. Yes, okay. So she will be talking on CLPRED, the sequence-based protein crystallization prediction using BLSTM neural networks. And can we see her presentation? Okay, she's sharing her screen. All right, go ahead.
Hello everyone, my name is Wenjing and I am a graduate student at Central South University in China. I'm very glad to be here and share our recent research with you all. The title of my oral presentation is CLPRED, a sequence-based protein crystallization predictor using BLSTM neural network. My talk consists of 4 parts. The first part is a brief introduction about protein crystallization. In the second part, I would like to introduce our material and methods. In the third part, I will show you the performance of our method. And lastly, I will present our discussions and then give a case study. First of all, I would like to introduce the background of protein crystallization. This is a dependency graph from protein crystal to drug design. Protein structure analysis is the base of protein functional research, disease treatment, and drug design. At present, about 80 to 90% of the non-three-dimensional protein structures are obtained using X-ray diffraction analysis technology. To determine the three-dimensional structures of proteins by X-ray diffraction, forming protein crystals suitable for structure analysis is crucial. Over 60% of the structure determination costs are consumed by unsuccessful attempts. Only about 2 to 10% of protein targets produce high-resolution protein structures, and about 4.6% of target produce diffraction quality. So if there is a prediction method that can predict the crystallinity of a protein with satisfactory accuracy, it will enable the rapid in silico screening of target proteins that are predicted to have lower crystallization propensity. And consequently, try and error settings and cost of the protein structure analysis will be significantly reduced. Next, I would like to describe our material and methods in detail. We have used 3 test sets to evaluate the performance of our method. The data sets we used are listed in this table. Before proposing our method, let me introduce the existing tools. Previous studies usually perform a 2-stage classification. performing feature selection and engineering using additional tools, and then feeding the selected features to the machine learning algorithm for classification. Recent work, Deep Crystal built a convolutional neural network to extract features such as frequency set of amino acid, k-mers, and k-mers information from the raw sequence. Studies in protein secondary structure show that the kinds of neighboring residues play a predominant role in the secondary structure conformation that a residue adopts. As we all know, CNN can often effectively capture local information such as local motif patterns between interaction of k-mers, but have difficulty in learning high-order long-range interaction of k-mers, which are essential to form stable spatial structures. Thus, mining the long-range peptide-to-peptide interaction in proteins is crucial to solve the riddle of protein crystallization propensity prediction. As we all know, recurrent neural network is usually effective in learning long-range interactions information. Long short-term memory is a special RNN designed to address the vanishing gradient problem, so that the network is able to learn over hundreds of time steps. Based on the above, we built a deep learning network to extract global interaction patterns, improving the precision over the traditional Windows-based neural network. This is the model architecture of our method. It is a deep learning framework, protein crystallization prediction only based on sequence information. It first takes a raw protein sequence as input and convert the amino acid into raw vector representation through a raw embedding layer. Then the raw vectors are passed to a CNN layer, whose goal is to capture the high frequency k-mers features. Finally, the k-mers features are supplied to the VLSTM layer, which capture the long-range interactions information between k-mers amino acid and generate predictions. Specifically, it uses feed-forward neural network for concatenation of prediction from the forward and the backward in the bidirectional model. Next, I will show you the performance of our method. We use multiple evaluation indicators, including the error under the curve, accuracy, recall, precision, F-score, and negative predictive value, Matthews correlation coefficient. We evaluate the predictive performances of these 5 sequence-based protein crystallization predictors by 10 flow cross-validation, as shown in the table. Our model outperforms the other predictors in terms of accuracy, AUC, recall, F-score, and PVMCC. But when measured by precision, our method is inferior to Deep Crystal. However, the gains of our model measured by other metrics are more significant. Here are the evaluation results of the other 2 data sets, as shown in the table. Our method usually obtains the best result compared with the other methods. In the last part, we will present our discussions and then give a case study of our methods. My first discussion is about model analysis. As shown in the picture, we project the feature vectors derived from each layer to a 2-dimensional picture space via TSNE, PCA, and UNAP tools. We can observe that as layers deepen, the learned feature vector become gradually more effective in distinguishing crystallizable proteins from the non-crystallizable ones. And secondly, by analyzing the hidden layer of the model, we have obtained the key amino acid that affect the crystallization of the protein. This is the top 7 amino acids that mostly contribute to the protein crystallization. It is generally consistent with the result present in ICM Christ, which was published in +1, while our result indicated that arginine and isoleucine are also important to protein crystallization. In order to demonstrate the effectiveness of BLSTM, we compare the prediction performances of CL-PRED with a new version which use CN layer instead of BLSTM layer. As shown in the table, we can find that for most of the matches, the prediction result of our methods without BLSTM and deep crystals are relatively similar. But the prediction result of our method with BLSTM is performed significantly better. This also confirms that the feature representation learned by ESTM plays an important role in effectively predicting which sequences can produce diffraction quality crystals. Crystallization prediction can be further improved when including additional features such as evolutional information and predicted protein properties such as secondary structure, flexibility, structure motif, and solvent accessibility. Recently, the neutral B crystal used pre-extracted evolutional, structural, physical, chemical characteristics as additional features and employ attributes to predict the protein crystallization propensity, which has shown significant performance enhancement. These features can also be used in our framework. The table shows the performance comparison among CL-PRED B crystal and CL-PRED AF, which combines the last layer features of CL-PRED with 641 additional features, including A-state secondary structure, fraction of adipose residues, disorder, and hydrophobicity. We can find that when these additional features are incorporated, the performance of CL-PRED AF is also significantly improved compared to the CL-PRED and is at least comparable to B crystal. Nevertheless, CL-PRED is as a predictor using sequence-only feature is still of practical use, since it does not rely on the accuracy of other feature prediction tools, and is not biased to the database of deriving additional features. At the end of my talk, I would like to give a case study of our method, which is Zika virus protein analysis. We have collected some crystallable Zika protein sequences and some other Zika proteins that no evidence shows that they can produce diffraction quality crystals. As shown in the table, our method is the only model that 100% correctly identifies the Zika virus protein which can produce diffraction quality crystals. We can also observe that the full length 2B NS2B is predicted with relatively high confidence by our method and 4 other tools, which deserve further investigation by Zika virus research community. I think that's all for my presentation. Thank you for your attention.
Thank you very much for your talk and for staying on time. We have 2 questions from the audience. One is, where did you get your data? Where is your data from?
I get the data from the previous studies, from the deep crystal and B crystal, then their data.
Okay, good. And can your tools be publicly used for free? So are they available for the public for free?
Available for the GitHub, and the GitHub is shown in my paper.
Okay. Okay, that's great. Thank you very much. So we are pretty much on time, so that's great. So we can move to the last talk of this morning by Janani Durairaj. something like that, from the Wageningen University. She's going to talk on geometricus that represents protein structures and assemers derived from moment invariants.
Yeah. Can you see my screen?
Yes.
Okay. Yeah, hello, I'm Janani. I'm from the bioinformatics group in Wageningen, and I'll be talking about geometricus, which is a new approach of representing three-dimensional protein structures. So first, why would we want a new representation? There's two aspects to this. One is that the number of experimentally solved structures has been steadily rising. We have over 1 million now. And at the same time, we are also getting a lot better at predicting structure from sequence using de novo methods or homology modeling. And because of this, we've started using more and more structural information in our analyses, for instance, in clustering or machine learning, to better understand protein function. And for these purposes, it's useful to have new representations. For geometricus, we took inspiration from the sequence world. So where Kamer counting is very common. Kamer counting is really simple. You take a sequence, you look at the overlapping subsequences, and you count how many times each unique subsequence appears. Because it's so simple, it's really fast, and it's used all over the place. So it's used in alignment and clustering, and even in classification. So how would you transfer this to the 3D world? So you have to transfer the two steps, the fragmentation step and the counting step. In terms of fragmentation, there are multiple ways in which you can fragment the structure. We looked at two. One is Kamer-based, so using the amino acid sequence, you look at contiguous overlapping Kamers, but in 3D. And then the radius-based approach, which takes each residue as the center of a sphere of a certain radius, and then considers all of the atoms within that sphere as belonging to the same fragment. So with these approaches, you get a set of structural fragments, but unlike with the 1D sequences, you can't immediately compare them because they could be in any location and any orientation. So it turns out we're not the only field to have had this problem. So also in computer vision and robotics, you have applications like character recognition or pose and gesture recognition, where you would want to compare different objects which are translated or rotated with respect to each other. And one solution for this that they've come up with is rotation invariant moments. And these are essentially mathematical formula that take as input the X, Y, and Z coordinates, perform some kind of manipulation, and return a number which is invariant to rotation, meaning you can rotate your coordinates and put them through the same formula and you'll get back the same number. So I showed two examples on the slide here, but there are 4 of these moment invariants, meaning that you end up with 4 numbers that describe different topologies or shapes of your structural fragments. We're still one step away from counting because these are 4 floating point numbers, so you can't exactly equate them. And to solve this, we just multiply it by a certain resolution, m, and then take the integer value. So here are 2 synthetic examples of structural fragments, and they're rotated in 3 different ways. And you can see that all 3 of these have the same set of moment invariants, and you could multiply them by 1 or by 2, for instance, and take the integer value. And you get this discrete unit that you can now compare within proteins and across proteins. And we call this the shapemer. Right, so let's move to real data. So we calculated shapemers across around 90,000 structures in the PDB with less than 40% sequence identity. And we used, Kamer approach with Kamer size of 16 and the radius approach with a radius of 10. And the first thing that we found is that the distribution of fragments across shapemers is not very uniform. So you have some shapemers which have just 1 fragment. So these are really unique topologies. And then you have some which have over 1 million because this is on the log scale. And to zoom in a bit on some examples, what we've done on the top here is just choose 1,000 random fragments and superpose them so that you can see that they actually do represent the same shape. And you see one of the most common ones is just an alpha helix, and then this is something that's a bit less common. And similarly for the radius approach, you have a common one being an alpha helix next to another one, and then a more rare one being something on the surface. And one thing you'll probably notice here is that these proteins which we're comparing are really different from each other, but because of this approach, we can compare parts of them. Right, so to recap, this is the whole process. You start with a set of proteins, you fragment them, you calculate the shapemer for each fragment at a certain resolution, and then finally you can count the number of times each shapemer appears, and that gives you a count vector or an embedding for each protein. And since none of this involves superposition or anything like that, it's extremely fast. Okay, so now that we have the embeddings, let's look at some applications of what we can do with them. You could compare proteins across families. So here is dimensionality reduction of proteins from 10 different PFAM domains. And you can see that it separates quite cleanly. But because of the versatility of the approach, you're not really restricted to comparing divergent proteins. So for instance, here we can zoom into this orange blob, which is the protein kinases, and look at a subfamily, the MAP kinases, which have 3 subtypes. And here's where the resolution parameter comes into play, because if you use a low resolution, then you don't get a good separation because these are really similar proteins and your embeddings are too coarse. But as you increase the resolution, you can suddenly separate them much cleaner. Another application is in structure similarity search. So on the right are just a few of the many topologies that proteins take. And in some applications, it's really useful to be able to find similar topologies to a query protein. For instance, if you want to look at different templates or you want to compare functions. And in this task, we benchmarked Geometricus and found that it was faster and more accurate than other alignment-free approaches. And unlike the others, it doesn't depend on a fragment library and it doesn't depend on a training set. So you can really easily and quickly adapt it to different tasks. And this gives something that's close to the alignment-based approaches, but much, much faster. And speaking of alignment, we also realized that we could use Geometricus in multiple structure alignment. So in the same way that sequence alignment tools like Clustal use k-mer counting to make like a guide tree, we can use shapemer counting for the same purpose and get something that's a really fast structure alignment tool. And finally, a really neat application, I think, for this is in interpretable machine learning. And here we'll go back to the MAP kinase example. So we have 3 different subtypes and we predict which one a protein belongs to with quite high accuracy. But what we can also do is use feature selection to select the predictive shapemers to pinpoint which shapemers are actually causing or are informative for distinguishing these 3 classes, and then map those back to the residues where they belong. And here I have an example of just this one red shapemer, which is present in almost all of the P38 kinases at least 2 times, and is not present in the other 2. And if you actually look in the structure, you can really clearly see which shape is causing this difference. And so this really means that you now have a new topological source of features that you can use by itself, or you can augment with existing sources like electrostatics or dynamics. And this could lead to new structure function relationships in protein families. Yeah, so I'd like to thank Mehmet, who's my co-author, and also in the audience, Barbara and Rens for their input, my supervisors, and the group at Wageningen, the bioinformatics group. And you can get Geometricus with pip. The code is open source at GitHub, and you can also find the documentation there. Thank you.
So thank you very much for staying on time. So we have 7 minutes. Wow. Okay, so we have time for questions. So there is first a question from the audience from Hoang Pham, who is asking, how does your method deal with the flexibility in surface loops?
Yeah, that's a really good question. Actually, right now it doesn't use flexibility, so it uses just the crystal structure coordinates. But that's something that we actually really want to look into, because you could imagine that you could use things like normal modal analysis and such to get the flexibility, and then maybe incorporate it into the moments as a probabilistic thing.
Okay, I have a question myself. I'm not a protein structure person, so maybe my question is a little bit naive. So you are trying to infer the shape for camels, for let's say small protein fragments. So I can imagine that some camels will be harder to shape than others. So is that an issue when you are then making your comparisons, your counting, I mean, your applications?
So if I understand correctly, you mean because in the end, these you're representing a set of coordinates with just 4 numbers, so you're definitely going to make some approximations there.
Yes.
Yeah.
So I'm wondering whether every shape is equally described, right?
Yeah, yeah. No, that's true. And this is something that right now we use this really simple approach of, multiplying it by resolution and taking the integer, but it could use some more exploration in terms of, like, you could try clustering the moments and seeing if one moment is more descriptive of certain kinds of shapes than others and so on. And that could definitely help, but it might increase the running times, of course.
Okay. Yeah. Right, more questions on the board. What length of window do you use for the Shamer by Diego Amaya Ramirez?
Yeah, so for the things that I presented today, it was a Kamer size of 16 and a radius of 10 angstroms. But yeah, this can be changed according to your application, of course.
Okay, thanks. So Jan Niklas has another question. How does the similarity search exactly work? What does the user query?
Okay, so this, the similarity search, I didn't really go into detail, I guess, but it's based on 2 classification schemes, the CAP and SCOP datasets, which classify protein topologies. And the idea is to, given a query protein, have the most similar one belonging to the same topology. And that's how you sort of benchmark whether your similarity search is working or not.
Okay.
Does that answer the question?
I hope so. And one last question by someone with a very difficult name to pronounce, who is asking of saying that this representation is very useful in differentiating domains. Have you tried with different domains which are closer?
Yeah, so for instance, the MAP kinase example are proteins which share a very similar structural fold. So there you would need a higher resolution, and that gives like a longer embedding, but in the end you get something that can distinguish really similar proteins. I mean, you could even use it on things like single mutants if you want, if you really increase the resolution.
Well, I guess since we have time and the questions are continuing popping up, we can go ahead with maybe one more, and then we will let you rest. Do you consider false when considering a group? Do you consider false when considering a group, from Shaila Verma?
I'm not sure I understand the question. What exactly do you mean by false?
So Shaila, can you be more specific on your question, please?
Maybe you mean in the structure similarity search with CAP and SCOP? If that's the case, then we go up to the topology level in CAP.
Okay, I'm not seeing a reply from her.
Yeah, you can definitely get in touch
you
with
can
me
definitely
later.
touch offline after this session. Okay, well, I want to thank everyone for being here, for staying on time. I think it has been really a great set of talks, and I think that yeah. So this is it for this session. We have now 20 minutes break before the next keynote speaker. So by 11:20, you can come back to your computer and to the platform for the next session. So thank you very much, and see you later.